Have you ever wondered about how scientists extract DNA from cells? DNA extraction is a fascinating process used for scientific research, medical diagnostic, and even forensic investigation. Today, we will walk you through the 5 key steps of this process. The first step of this process is cell lysis. This step is to break open the cell and release the DNA. A lysis buffer containing detergent is added to digest the cell membrane. The step is followed by separation. In order to remove the protein and impurities, an enzyme called proteinase K is added to digest the protein, leaving the free DNA in the solution. After that, we do precipitation to isolate and concentrate the DNA by adding alcohol. DNA is then clamped together into a visible white layer. Layer is separated by centrifugation to obtain the DNA pellet. After that, we do purification. The DNA pellet is washed by a 70% alcohol to remove the remaining impurity. Once the cleaning process is done, the pellet is air drying. The last step is resuspension. Finally, the dry DNA pellet is rehydrated using water. This ensures the DNA is preserved and ready for future use. And there you have it, the journey of DNA extraction, from a cells to a purified DNA.
Now, let's compare two widely used techniques, the traditional phenol chloroform methods and the modern commercial kit approach.
DNA extraction is a method to purify DNA by using physical and chemical method from a sample, separating DNA from cell membrane, protein, and other cellular component. The similarity of both methods is to separate DNA from other cellular component and contaminants. While both achieve this, they differ significantly in terms of DNA purity, concentration, time efficiency, and cost.
Let's begin with DNA purity. Using the phenol chloroform methods, only 26.7% of sample contain pure DNA. In contrast, the commercial kit yield pure DNA in 80% of sample. This makes the commercial kit a clear winner for application requiring high DNA purity. When it comes to DNA concentration, the phenol chloroform methods produce a higher DNA yield compared to commercial kit, making it a better choice for studies requiring large amounts of DNA.
However, in term time efficiency, the phenol chloroform method is more time-consuming and less convenient. Meanwhile, the commercial kit is designed for less time-consuming and more convenient, which helps saving researcher valuable time in the lab. Finally, let's reconsider the cost for DNA extraction. Hence, the phenol chloroform method is less expensive, making it more accessible for labs with limited budget. Nevertheless, the commercial kit is more convenient and comes at a higher cost. In summary, the phenol chloroform method provides higher DNA yield at a lower cost but sacrifices time, efficiency, and purity. On top of that, the commercial kit offers a greater purity and convenience, making it ideal for time-sensitive projects even though at a higher cost.
Next, we talk about the importance of the DNA purity. DNA purity is important to ensure the reliability and accuracy of biomedical research. First of all, DNA purity is one of the key factors contributing to high-quality DNA. It ensures the utility and long-term availability of DNA generated. DNA purity is closely related to downstream applications such as polymerase chain reaction (PCR) and the availability of the DNA source for the research. Therefore, high DNA purity can lead to reliable research in the downstream application. Besides, high purity DNA allows for precise quantification and qualification. Without pure DNA, results can be unreliable, leading to low confidence findings and wasted resources, which include cost and sample. Furthermore, high DNA purity leads to reliable measurement of DNA concentration for molecular biology application. Contaminants present in DNA sample reduce DNA purity. Contaminants such as dust particles, chemicals, or RNA can distort measurements, especially during spectrophotometric analysis, by interfering with light absorption at critical wavelength. These chemicals cause an increase in the extent of absorption. Hence, low DNA purity leads to unreliable measurement and poor quality results.
To assess DNA purity, spectrophotometry is used along with the absorbance ratio at 260 and 280 nanometer. Absorbance at 260, 280 and 230 nanometer was measured in duplicate photomicrolith of each DNA sample using the nanoquant plate. DNA is considered pure if the 260/280 ratio is approximately 1.8. Ratios below or equal to 1.6 may indicate the presence of proteins, phenol, or other contaminants that absorb near 280 nanometer. The percentage of sample in each group with a purity ratio between 1.6 and 2.0 was also calculated. The 260/230 ratio serves as the secondary measure of DNA purity. For each sample, the 260/230 ratio was estimated and compared to DNA concentration. Pure DNA typically has a 260/230 ratio between 2.0 and 2.2. Ratios lower than expected may indicate contaminants that absorb at 230 nanometer. However, the 260/230 ratio is an unreliable DNA quality indicator when a saline elution buffer is used, as salt concentration increase more than DNA concentrations in such cases. This caused less concentrated samples, despite equal purity, to show lower 260/230 ratios due to the salt absorbance at 230 nanometer.